Hi, my name is Brandie White, and I'm a first-year PhD student at SDSU, rotating in the Rower Lab, and I'm interested in how microbial communities are structured and communicate with each other. One of the molecular mechanisms of this is vesicular communication. So, no cell is an island. Whether you're a multicellular organism or a unicellular organism, you always interact with your environment and the other members of the environment. So, for this talk, I'm going to focus on a really simplified bacterial cell. This is the membrane. This is the bilipid membrane here. Here's your cytoplasm. And vesicles are made by blebbing off of the membrane surface and forming a sphere that contains cytoplasm and often specific proteins and molecules that are meant to be ejected from the cell, and also specific markers in the membrane. Now, when these were first found, nobody knew what they were. And everybody assumed maybe this is just some way of getting rid of cellular waste. And they weren't paid a lot of attention. But when Thomas Südhoff was studying synapses, he discovered that vesicular trafficking was responsible for transmitting signals in the brain, and he won a Nobel Prize for this. This renewed interest in learning more about vesicles and what their function is. So what I want to talk about today is some of the functions of bacterial vesicles. And one of these functions is communication, such as quorum sensing. So even if two bacteria are not in physical contact with each other, by the production of vesicles binding to sensors, they can tell that they are in proximity. And this is important for initiating certain pathogenic behaviors, such as biofilm formation. Another important feature is cross-feeding. So one bacterial cell emits some vesicles, and if another consumes it or absorbs it, they can have either positive impacts if it's a food or nutrient that that bacteria needs, or if it's a piece of DNA that offers an evolutionary advantage, or it can be negative if it has toxins inside. And a third very interesting function of vesicles is in protection against bacteriophage. So when the bacteriophage goes to infect the bacterial cell, if it has covered its surface with vesicles, the bacteriophage might inject its DNA into a vesicle instead, leaving the bacteria unscathed. So this is what I'm interested in studying from my research. And I focus on mucosal surfaces because they're enriched for bacteria, and they're found in a variety of places, such as the human gut, and also in the environment, on the epithelium of marine creatures, such as sharks and corals, both of which are endangered. So by studying and isolating vesicles from these communities, I can say, who is there, what are they producing, what is inside these vesicles, and how do they function? And having more answers to this can have excellent widespread impacts in diagnostics, for health status, as well as targeted drug delivery, which might serve to produce synthetic vesicles that contain medicines that are beneficial to promoting a healthy community of microbes. Thank you very much.
Thanks for watching another presentation here at the Science Class. If you want to know more about the research that you just heard about, check the video description below for a link to that lab's website. And if you want to see more presentations like this in the future, You can subscribe to us by clicking this button here. Thanks very much.